Bismillah Rahman Rahim. Chemotherapy or antibiotics are the very important unit of pharmacology, and you can say it is backbone of pharmacology. What is chemotherapy? Chemotherapy is the drug treatment for the diseases caused by pathogenic microorganisms, bacteria, parasites, and tumor cells. Chemotherapy is the drug treatment for the diseases caused by pathogenic microorganisms, bacteria, parasites, and tumor cells. What are antibacterial agents or drugs? These are the agents used to prevent bacterial infection by killing or preventing the growth of the bacteria. Antibiotics have no role on viral infections mainly. How do you classify antibiotics or any drug? Antibiotics can be classified on basis of chemical structure, on basis of source of drug or origin of drug, can be classified on the basis of spectral activity or range activity of the antibiotics, whether they are narrow spectrum or broad spectrum, or on the basis of mechanism of action, or antibiotics can be classified on basis of whether they are bacteriostatic or they are bactericidal. Antibiotics can be classified on the basis of their pharmacological actions, their pharmacological activity, and their pharmacological effects. Antibiotics can also be classified on the basis of routes of drug administration. So, these are the various ways to classify the antibiotics. The main classes of antibiotics or types of antibiotics are beta-lactams, macrolides, quinolones, tetracyclines, aminoglycosides. Now, what is antibacterial agent? These are the agents which are used to prevent the bacterial infection by killing or preventing the growth of the bacteria. Common terms used are bactericidal, bacteriostatic, minimal inhibitory concentration MIC, minimal bactericidal concentration MBC. So, bactericidal, you should be clear, it is defined as a chemical agent capable of killing bacteria, destroying the bacteria, but not necessarily bacterial spores. On the other hand, other type of antibiotics are bacteriostatic. The word static, it is defined as the chemical agent capable of preventing the growth of the bacteria but not destroying or killing the bacteria. Here reproduction or replication is prevented. While in bactericidal there is killing of bacteria, in bacteriostatic type of antibiotic there is prevention of the growth of the bacteria. So, chemotherapy, example of bacteriostatic antibiotics and then the example of bactericidal antibiotics. We take first bactericidal antibiotics. They are bacteriostatic. They are tetracycline, group of tetracycline antibiotic, chloramphenicol antibiotics, erythromycin, clindamycin, sulfonamides, trimethoprim. On the other hand, the antibiotics which are bactericidal, killing the bacteria, destroying the bacteria, are aminoglycosides, beta-lactams, vancomycin, quinolones, rifampicin, metronidazole. You should also be knowing that antibiotic was abstracted from the word antibiosis, which means against life. Antibiotics may be classified, subclassified as antibacterials, antifungals, antivirals. So the drugs acting against bacteria are antibacterial, against fungi or fungus are antifungals, and the drugs acting against viruses are called antiviral drugs or antivirals. As far as mechanism of antibiotic is concerned, it may be inhibiting cell wall synthesis. This is the mechanism of antibiotics, mechanism of action of antibiotics. On basis of inhibiting cell wall synthesis, you have grouped antibiotics under the heading the drugs which inhibit the cell wall synthesis. And the other group is antibiotics which inhibit the protein synthesis. And the third group is the drugs or antibiotics, those inhibit the nucleic acid synthesis or their function. So, under the heading of inhibiting the cell wall synthesis, we include beta lactams as penicillins, as cephalosporins, carbapenems, monobactams, vancomycin, daptomycin, and polypeptides. Rest of the drugs or antibiotics are present in the group of protein synthesis inhibitors and inhibiting nucleic acid synthesis. And now I'm not going in detail of those, as I am concerned here with the antibiotics inhibiting the cell wall synthesis, that is beta lactams, penicillin, cephalosporin. So these are the antibiotics which inhibit the cell wall synthesis, that is penicillins, cephalosporins, carbapenems, glycopeptides. Now I come to the main topic of antibiotic, that is cephalosporins. What is the structure of cephalosporins? Cephalosporins are bactericidal drugs which destroy or kill the bacteria, so useful in bacterial infection, fruitful in bacterial infections. The cephalosporins are a class of beta-lactam antibiotics originally derived from fungus Acremonium, known as Cephalosporium. The cephalosporins are called beta-lactams. Why? Because of the unusual four members beta-lactam ring that is common to all their members of the drugs, like present this ring, beta-lactam ring in penicillins. This beta-lactam ring is present in cephalosporins. As you can see, the structure, chemical structure of the penicillin, and then you can see the picture of or structure of the cephalosporin beta-lactam rings present in both of them, penicillins and in cephalosporins beta-lactam ring. Then what is cephalosporins? Cephalosporins antibiotics are bacterial cell wall synthesis inhibitors. The cell wall is composed of polymer, consists of glycan units joined by peptide cross links, making beta-lactam ring structures. Cephalosporins are beta-lactam antibiotics closely resembling structurally as well as functionally to the group of penicillins antibiotics. So cephalosporins have same mechanism of action or mode of action as that of penicillins. Cephalosporins tend to be more resistant to beta-lactam enzymes than penicillin. What is the mechanism of action of cephalosporins? As I have told already, it is the mechanism of action. Its mechanism of action is like penicillins. Cephalosporins inhibit bacterial cell wall synthesis by binding to beta-lactam ring present on bacterial cell wall. This results in inhibition of cell wall synthesis by blocking transpeptidation of bacterial cell wall peptidoglycan by interfering with the enzymes transpeptidase and peptidase. Most cephalosporins are produced semisynthetically by the chemical attachment of the side chains.